in this talk, I'm going to focus on the chlorophyll synthesis. Okay, and as I talked to you before, the greening of plants, the vascular plants in general, if you grow in the dark, it becomes etiolated, and then if you grow them in the light, it becomes green, fully green. So here, etioplast, cell contain etioplast, and here it's chloroplast. So I introduced Chlamydomonas greening, and the Chlamydomonas is, as I told you, that accumulate chlorophylls when grown in even in the dark, okay, because it contains light-independent protochlorophyll reductase. So it can synthesize, the cell Chlamydomonas can synthesize chlorophylls in the dark. But if you illuminate the cell, of course, synthesis rate of chlorophyll enhanced. So the cell becomes more green, but you can grow the green cells in the dark. But I talked before, the Y1 mutant is yellow in the dark mutant, like a vascular plant. And this mutant cannot synthesize chlorophyll in the dark because light-independent protochlorophyll oxidoreductase was deficient. It's deficient. So only light-dependent oxidoreductase is present. So if you grow the cell in the dark, it becomes a yellow cell, and the chloroplast become etioplast. Okay, and this advantage of Chlamydomonas is if the dark grown cell is shifted under the condition of light, it starts to synthesize chlorophyll, and then about 8 hours, it becomes fully green. It's okay for okay. Okay, so so we can follow the process during a greening using this Y1 mutant of Chlamydomonas. Okay, so I showed before, but just remind you of the process. So from zero, dark green, dark grown Y1 cells, yellow cells, and then greening, you extract the pigments, and then measure the absorption spectra. Then 0 hours, there's no chlorophyll. Okay. Okay. This is convenient. Like this. You can see better. No chlorophyll accumulation of carotenoid. And during illumination, the chlorophyll accumulate. Okay. Like this. Okay. And chlorophyll binding protein like photosystem 1 and photosystem 2 and LSG, doing a greening, yellow cells, you don't see these chlorophyll binding proteins. But during a greening process, PS1 accumulate, PSAA, these are all PS. LSC1, PS2, LSC2 accumulate. Although I told you that the cytochrome B6F complex contains one chlorophyll molecule, but this is PET A is cytochrome F. Although the amount is lower, but still accumulating even in the yellow cell. That is an interesting point. We have not characterized further, but in the absence of chlorophyll, B6F is still at a reduced level, but it accumulate. They increase it like this. Okay, that's interesting point. So how the pigment accumulation occur? So this is just absorption. A mixture of chlorohyls and carotenoids. We don't know which component accumulating. So we analyzed pigments by HPLC. We extract the pigments with organic solvent and analyze composition of pigments by HPLC. This is the elution time and also absorption 270 nanometer, 430 nanometer, 43 nanometer, depending on the component absorption peak are different. So you can choose the detection wavelengths. This is an example. For example, 30 is you can detect the chlorohyls and the carotenoids. So this is the elution time. So first hydrophilic xanthophyls, rhodozanthine. This is a criminomerase. So rhodozanthine, neoxanthine, biogenesis, lutein, and chlorophyll B, chlorophyll A, alpha, beta carotene are separated like this. Okay. And if you want to analyze the quinone, 247 nanometer, you can detect a very small peak of quinone here. And naphthoquinone, you can detect here. In the craminomerase, hydroxyphyroquinone is present in PS1. This is indicate the measure of PS1 content because this quinone present in photosystem 1. And plus quinone is present in cytochrome membrane, bilayer, lipid bilayer, and photosystem 2, both. And during illumination, we measure this component. Okay, the chlorophyll A and B, you can detect here chlorophyll B and chlorophyll A. It's a huge peak. It's easy to detect. So during illumination, chlorophyll A increases like this, and chlorophyll B like this, up and down. And hydroxyphyroquinone, this is associated to PS photosystem 1. It's increasing. Oh. Like this, and plus quinone going up like this. And this is the carotenoid. Alpha carotene, the beta carotene is a carotene without oxygen atom, so carotenes. And lutein, rhodozanthine, biogenesis, neoxanthine is the xanthophyls containing oxygen atoms. So this is a little bit more hydrophilic compared to this carotene. That's why the alpha and beta carotene eluted later than xanthophyls. So this is the changes in this carotene. Okay, rhodozanthine is somehow after lag phase it increases, but the beta carotene decreases, and biogenesis increases, and alpha carotene almost constant. Rhodozanthine, neoxanthine, xanthophyls are increasing. Okay, these are mostly associated with light harvesting complex. So probably this reflects to accumulation of light harvesting complex. However, although here chlorophyll A and B are absent, almost absent, but if we look at carefully, this is after 5 hours greening cells, we detected many chlorophyll-like peak by HPLC profile. See here. Chlorophyll A is here. Chlorophyll B is here. Okay. And chlorophyll A type peak A1, A2, A3 were also separated before the chlorophyll A separation. Okay. And chlorophyll B, in addition to chlorophyll B, chlorophyll B1, B2, B3 were separated somehow. And if you look at the absorption spectra of this, chlorophyll A, A1, A2, like that, the absorption spectra was exactly the same. Okay, chlorophyll B type also the same. There's no indication that A1, B1, B2, B1, B2, probably this is B3. Okay, the same. But in the separated in a different position. That's happened so well. So protochlorophyll A is converted to chlorophyllite, and then converted to chlorophyll A and chlorophyll B. But there are additional steps. This is called chlorophyll A GG, chlorophyll A DHGG, chlorophyll A THGG, and then chlorophyll A. So this is a abbreviation of, this is geranyl chlorophyll A. And dihydrogeranyl chlorophyll A. Tetrahydrogeranyl chlorophyll A. And the same series of chlorophyll B type also present. So this is A1, corresponds to A1 here. And then the dihydrogeranyl chlorophyll A is here. And then tetrahydroxy geranyl chlorophyll A is here. And then finally chlorophyll A. So this is a stepwise conversion tool. So this usually in the white type cells, this component you could detect small amount, negligible amount. So usually we don't care very much, but doing a greening experiment, so it's starting from here, so it's substantial amount of this intermediate or derivative of chlorophyll A and B accumulate in during the greening cell. Okay, this happened. So geranyl reductase is involved in this conversion. This is also geranyl chlorophyll is this is a phytol, but geranyl chlorophyll A has additional this double bond here. So geranyl reductase stepwise this double bond is hydrolation, adding a hydrogen atom to remove the double bond. Then finally, only one double bond remained in a phytol chlorophyll A. So 1, 2, 3 steps, okay, occur. Also, this enzyme also involved in alpha tocopherol synthesis. This is also geranyl. Okay. Sorry. Also, naptoquinone, menaquinone, vitamin K, pyroquinone, pyroquinone, also the same structure is also not reduced. These are involved. But now we focus on this part. So there's a, in parallel, this geranyl reductase occur in a geranyl. This is a precursor of phytol. The geranyl is here. The geranyl portion to connect it. So that's why this is geranyl called. And pyrophosphate. So this is a phosphate. So the granular reductase is CHLP gene or proteins. This is CHLP gene encodes this enzyme. And this also granular pyrophosphate is reduced to phytol pyrophosphate. This is phytol. Okay. So this acts in this process, and this is chlorophyll A, and this is bind to chlorophyll A. This phytol may bind to phytol to make chlorophyll A, or granular pyrophosphate reacts with chlorophyllide A, and then to form chlorophyll A, granular chlorophyll A. And then this is reduced to chlorophyll A. So that two pathways occur. It is proposed that two pathways occurs. Okay, we don't in our this greening experiment we cannot distinguish between these two pathways. So we just make a graphs of this our results during a greening. This happens that the greening yellow cells, and this is fully green cells. So chlorophyll A is accumulated like this. There's a lag phase here, 1 to 2 hours. So in the beginning of 2 hours, almost no chlorophyll A and B accumulate, and then start to increase. But before the chlorophyll A accumulation, granular chlorophyll A starts to increase, and the peaking at 3 hours later of the illumination, and then slowly going down. And then the granular chlorophyll A increases a little later, and then peaking at 5 hours, and then going down. And tetrahydrogranular chlorophyll A also going up, and then peaking at 5 or 6 hours, and then going down. Oh. Similar pattern was observed for chlorophyll B derivatives. Okay, so this happens. So doing a greening, we cannot say very clearly, but it's a greening. This process occurs. Okay, and also chlorophyll B is we don't know exactly. The chlorophyll B is usually synthesized from chlorophyll A, but here even granular chlorophyll A is converted to chlorophyll B, granular chlorophyll B, and this reaction also occur, and also this reaction also occur. So this is happening. This mutant. So just want to check. Okay. So doing a greening in at least this chlamydomonas Y1 mutant, the accumulation pattern of chlorophylls are a little bit different from the wild type, and abnormally this chlorophyll A derivative and chlorophyll B derivatives accumulate transiently. This happens. Okay. Then we analyzed chlorophyll binding proteins. It's gradually accumulated, as shown in the beginning of the Western blotting. But here we show the complex, not apoprotein accumulation, but complex accumulation is analyzed. Not every time, because every hour, because it was so hard. So we tested 2 hours and 10 hours after 10 hour, 2 hours, and 10-hour illumination. This is very initial illumination. So it's very low amount of chlorophyll accumulated, and this is free wind. Okay. So here, the most cells are still yellow, so it was not easy to accumulate the cytochrome membrane from these cells. So we have to start from the plant to huge amount of culture. And then we collected the membrane, break the cells and collected the membrane fraction and solubilized and separated by successive gradient ultracentrifugation, as I showed many times already. So this is a gradient of top of the gradient and 0.1 molar sucrose, and this is the bottom of the gradient and 1.3 molar sucrose. So smaller complex separated here, and the larger complex separated here. So you see the difference. We try to load the similar amount of the chlorophyll, but obviously this is less. But the pattern looks different. It's not the same. See? So this is we fractionated this gradient from the bottom, fraction number, and then we took the absorption of the fraction. So this is a 675 nanometer. That means chlorophyll peak, chlorophyll absorption. Okay, so from the 10 hours, it's a typical pattern like Y type. A1, A2, A3. This is LHC2, PS2, or PS1 core. And this is PS1 LHC1 super complex. But in a 2 hours illumination, the complex formation was incomplete. So we could detect A1, A2, a very small amount of A3. See, only. We call this peak, this is shoulder. Okay, after peak, it's the shoulder. And then we analyzed all the fraction, analyzed the polypeptide from all the fraction, and we detected this is immunoblotting because the protein was not sufficient. So this is the 10 hours and the 2 hours. Okay, only this part, right? So 10 hours, the A3 fraction was here, PS1, a little bit PS1 in A2, and PS1, LHC1. We didn't detect the PS2 antibody, but this is obvious PS2. And also in 2 hours, already PS1 is accumulated, but the separated mainly A2 fraction. Here, okay, here. Now, after 10 hours, you detect the signal in A3, but here A2. And also here, PSA-A, D, G, LHC1 was mostly separated at the same position. But here, small amount of PS1 is present in A3, and PSAG is here. But A2 major PS1 was separated in A2, and the PSAG is here, but PSAG was not here. And LHC5 was a little bit separated very broadly. We couldn't detect only small amount. It was signal was so weak we couldn't detect. But probably PSA5 also present here, but signal was so weak. So A2 here, A3 here. Then we analyzed the pigment in this A3, A2, A1 fraction. Okay, this is not the total. This is a pigment composition in each chlorophyll protein complexes. A1, A2, A3. And also doing illumination, how it changes. So chlorophyll A is as expected, chlorophyll A increases. Doing illumination in A1 fraction, chlorophyll A increases. And granular chlorophyll A, this is the first one, go down. And chlorophyll dihydrogranular chlorophyll A go up and transiently go up and down. Tetrahydrogranular chlorophyll A is also things. Okay, the pattern is very similar. H fraction, this is PS1 core and PS2 core, also the same. And H3 also the same, very similar pattern. So each chlorophyll protein complexes associate in doing intermediate illumination, associate both chlorophyll A and chlorophyll A derivatives. And chlorophyll B also the same, very similar. So this suggests that chlorophyll A, not only chlorophyll A and chlorophyll B, but granular chlorophyll A and B are integrated into chlorophyll protein complexes without waiting. No, the granular chlorophyll A is converted to chlorophyll A because these are very hydrophobic, so it's already integrated. So immediately this synthesized, but it's converted to chlorophyll A in the chlorophyll protein complexes, right? So somehow the granular reductase convert these derivatives present in the chlorophyll protein complexes. So we suspected that some pigments are present inside the protein complexes. So enzyme granular reductase may not access to the these granular chlorophylls, but it's actually it seems that it's access to in the complex. Okay, so that's the. I think I didn't put. Okay. So this is a chlorophyll synthesis, but this is the very last steps of the chlorophyll synthesis. One of the last steps of chlorophyll synthesis is the binding of the phytol or granular chlorophyll to chlorophyllite. So these steps makes pigments hydrophobic. So this cannot be chlorophyll cannot be present in soluble state. So it must associate into chlorophyll binding protein to form complex. So we expected this process may be somehow coupled with chlorophyll protein complex assembly. But so far we haven't clear data that show that it's coupling. So I didn't include in this slide. Maybe next lecture we are going to talk about the mutant of this granule reductase deficient mutant. I didn't prepare today, but the next lecture probably I can talk to. So this is also we wanted to try to analyze the molecular mechanism that couples the subunit assembly and cofactor assembly, the chlorohyl assembly, how it works. So probably this the chlorohyl synthesis the last steps of the chlorohyl synthesis must be coupled with this assembly process. But so far we haven't obtained the positive data showing this coupling for the moment. And also we next lecture I'm going to show the this granule reductase deficient phenotype, but still chlorohyl protein complex are assembled. So far it's difficult to analyze. Okay, so this is just greening and detailed greening process. So I just add some additional data again about the assembly of PS1 complexes. I show you this data before, and how do you say? I didn't tell you a detail, so I show you detail now. This is a separation. This is Y type. Okay, the separation pattern of chlorohyl protein complexes that you saw many times already. LHC2, PS2, and PS1 LH1 supercomplex was separated like this. And we separated these chlorohyl protein complexes from the cells knowing logarithmic phase. Logarithmic phase means the cell is dividing very actively. That means the cell synthesizes proteins actively. So, for example, PS1 proteins are actively synthesized. Okay, the activity of the assembly is active. Also, this is separated from the cells. That was the growth of each growth is stationary phase. So it doesn't divide anymore, but still cell is alive, but do not synthesize the proteins actively. It's very protein synthesis activity is very low. So PS1 assembly is very low, almost stopped. Okay, and we separate it. Looks very similar. Okay, it's not interesting, but we fractionate it. Okay, and the peak of chlorohyls was addition pattern. It shows A1 and A3 is almost the same, but we see a slight difference in the intensity of the H band. If you look at here, you can see the difference. Here, it maybe had a little bit weaker. You see, this small change was very important. Then we fractionated this. Okay, this is LHC2. You can see 25 to 30 kilodalton protein here. This is type 1, type 3, type 4 LHC2 apoprotein. This is PS2 protein. And this is PS1 protein. Okay, major, minor, small subunit. And LHC1. And we very much carefully analyze the polypeptide pattern by immunoblotting. This is staining, but this is immunoblotting. Okay. So from the logarithmic phase, the cell from the logarithmic phase you see a PS1 here, A3 band. Here. Right? Strong band. All the subunits and LSU1. But this is a A2 is PS2 proteins here. Okay. But if you look at carefully, there's a minor amount of PS1 is observed here in A2. Not PSA G and K. Okay. G and K I talked before. So this is the same, the difference. So we thought two possibilities from this data. Okay. One is these are just when we solarize thyroid membrane artificially, some of the subunit was dropped off, and then a smaller complex was generated artificially. So this fraction was because of the detergent. PS1 complex was becomes subcomplexes, smaller complexes. So it doesn't mean anything. But if you look at the separation pattern from the cell from the stationary phase, we didn't detect any minor PS1 here. So this suggests that, this could be present only the cells that the actively synthesize photosynthesis apparatus. So suggesting that this could be assembly intermediate. Usually we don't detect because assembly process proceed very rapidly. Okay. So I showed before that this pulse test labeling, and I didn't explain detail. So if this is not artifact, this is a newly synthesized subcomplex in the intermediate subcomplex. After pulse labeling, the protein PS1 here may not be labeled, but here it's labeled more strongly than others. That is a hypothesis before we start the experiment. Okay, so this is the result of pulse labeling. Because for the radioisotope work, we used a very small scale experiment. So this is after pulse labeling and chase experiment. We separated by sugar centigrade the chlorophyll protein complex, but using a very small tube. No, we wanted to use a large tube like cold experiment, but it was not possible because we have to work in a separated facility to handle with a radioisotope. So there we cannot use a large centrifuge. And also amount of sample, we have to work in a small scale of sample to somehow. If we use a large, so we have to use a lot of radioisotope and expensive and dangerous or something like that. So small. So separation was not so good as before, but we could separate, A2, A3, A1, A2, A3 band like this. It's a little bit more overlapping, but we could separate. Okay. So after pulse labeling, it's only 1 minute. It's very you have to work so carefully. And then labeling is this is PS1 reaction center. Was mainly detected in only A2, no smaller fraction. We couldn't detect. We barely detect signal in A3. Okay, and also some minor PS1 subunits are also present in A2, not A3, not much. Okay, and also this is PS2. As I told you, this is PS2 D1 protein that turn over very rapidly. Was present in not only A2 but also here in the upper phase. So it's not properly assembled proteins are present because this is a very strongly labeled turning over. So signal is so strong. So this is LHC2. Okay. And after chase experiment, it's difficult to say, but it's this PS1 protein shifted to A3 mostly. Separation was so delicate, but it shifted. If you look at the PS2, signal is here, peak is here. No, the peak of PS2 and the PS1 was the same here, but this is a little bit shifted. But there's a strong signal here, but this is sometimes aggregation of PS2 is migrated like this. So, but still, some are still remaining here, but it's shifted, obviously. Okay, right? Here, no minor PS1 stability is almost missing. It's shifted. LHC2 is more abundant because it took some time to integrate into the thyroid membrane because it translated in the cytosol, translocated into thyroid chloroplast, and then translocated to the thyroid membrane. It took some time, so it delayed. So, okay, this is the H3 band. So PS1 subcomplex detected in H2 is assembly intermediate. So it's not artifact. It's assembly intermediate. So we couldn't detect much assembly intermediate, but only assembly intermediate of PS1 core lacking LHC1 and PSAG and PSAK was right here. This here. And then this is H2. Then H3, LHC2, LHC1 is associated and PSAG and K associated to fully assembled PS1. You see, this is consistent with here, G and K are lacking, deficient. And the LHC1 is very low. So it's before the LHC1 and PSAG and K assembled PS1 core subcomplex accumulate. So this is only assembly intermediate we could detect. Because this step, the following steps, this following step is some rate limiting slow. So transiently, this subcomplex accumulate as a detectable level. Okay. So that's it. This is the same as no greening experiment, right? PS1 is first accumulated A2 band. So this independent experiment indicate the same lead us to the same conclusion. So probably this data can be reliable. Okay, so probably I stop my talk here and then ask you the question. I told the previous lecture that look at the data very carefully and speculate what we can extract from. So you don't have much time to see the data, so it's difficult for you to say something from data, but just by asking and hearing an answer, your thinking will be activated. So it's important not to listen to the lecture quietly, but asking even a stupid question is okay. Just talking stimulate your thinking. Okay, so I stop my talk and then ask you some question and comments or welcome. And then, is that okay? Okay, Sam, I ask you, how do you think it's the greening, there's some the derivative of chlorophyll, intermediate, accumulate. Do you have any question about this? No? What happened to the plant or algae if accumulate is not chlorophyll, not only chlorophyll A and B, intermediate accumulates? Gives you any questions? Maybe. No, this is a hint to ask a question.
Higher level of intermediates, they may be.
Sorry, say.
Higher level of intermediates of this pathway, they may be toxic to the plant.
That
they may be toxic to the plant.
Toxic plant. Okay, the question comes. So this accumulation, if you think that this intermediate are not functional, has no proper function as chlorophylls, that could be toxic under illumination, right? That question you didn't have? Okay. So far we don't know, but I'm going to talk later about this mutant that accumulate only granule chlorophylls. That analysis of this mutant gives us answer.
Are those steps compulsory? Without those steps also can we get chlorophyll?
Sorry.
Are these 3 steps compulsory, or if one step is missing in between those 6, will we get the?
If the GGR is missing, so the no chlorophyll A and chlorophyll B accumulate.
We knock it off. Or
knock out. Yeah, of course. This process is blocked, so only chlorophyll A. Granule chlorophyll A, and probably granule chlorophyll B accumulate, but no other. That is the case. So maybe you may have a question that granule chlorophyll A, does this granule chlorophyll A or B functions to collect light energy? Or in the case of reaction center, the chlorophyll A is involved in the electron transfer, charge separation. So granule chlorophyll A whether it acts as electron transfer component, primary electron donor or primary electron acceptor, no, redox reaction happens with this granule chlorophyll? That is the question, right? Another question is probably the granule chlorophyll A has 3 additional double bond here compared to chlorophyll, the phyto. No, this is only here. Double bond is here. So double bond mean this double bond is not flexible, right? So shape is rather fixed, but phyto is more flexible, right? So if these chlorophylls accommodate into chlorophyll protein complexes inside the proteins, right? So this is very like a rod, it's rigid. This is very flexible. So to accommodate into the proteins, I think this should have advantage. No, it's if you accommodate in some specific place, and this part disturbs the proper binding, then this can be bent or form flexibly, but this doesn't. So that prevent the proper binding. That's possible, right? You can speculate. So, but somehow the answer is there. Chlorophyll protein complexes are present. That is one of the reasons why we did this experiment, is that granular chlorophyll A prevent the proper assembly of chlorophyll protein complex or not. It seems to be working. So chlorophyll protein complexes structure is rather flexible somehow. More modification of the structure of pigments did not affect assembly. So the function, how about function? That is we discuss later, not today. So just keep it interested.
One question. So GGR, it is involved in the vitamin K tocopherol biosynthesis also.
Yeah.
So what happens in the middle? That also get affected, or?
Since the detection of the vitamin K phytoquinone was difficult, so this time we didn't analyze carefully. Because to be honest, from the beginning of our experiment, we didn't think about others. We just focused on only chlorophylls. And then we noticed, but it was a little bit too late because the student working this was graduated. So, but PS1 is assembled anyway. So, okay. So other questions? Okay. It's always have questions, but. Say it again.
This different chemical reactions are happening in same place or in different distributed, because they are using same enzymes.
GGR, yeah, these are
same enzyme.
GGR, granule reductase.
Synthesis also in localized in same place or in different place.
GGR.
Yeah.
Granule. We haven't tested, but at least a GGR, granule reductase, is present on the thyroid membrane.
Oh, other experiment also going on there only. Other alpha token.
These are all very hydrophobic molecules, so these are not present stably in the soluble part. So it should be present associated with the membrane protein or lipid bilayer. So at least a vitamin K1 or vitamin K1 or phytoquinone associated with PS1 in the middle of the hydrophobic region. And tocopherol is, I'm not quite sure, tocopherol. Maybe in the membrane. And of course, all chlorophylls are associated with proteins. No free, practically no free chlorophylls, because that free chlorophyll makes reactive oxygen species under the light. That's so toxic, so it's very dangerous. So GGR works as I showed that in the reducing granule chlorophyll present in the complex chlorophyll protein complex. So this is consistent that if GGR is present on the thyroid membrane, this is consistent, right? Data. Any other questions? Okay.
So what are the two pathways involved? You told two pathways are involved in also. You told two pathways are two pathways.
Two paths or what?
You told there are two pathways involved in this chlorophyll A and chlorophyll B. That cycle you told.
This one? Or path, the same enzyme?
Two pathways.
Two paths.
This is one pathway, this is one pathway.
This one pathway? Okay. Both way. This way or before it's associated, granule phosphate is before associate here also. We don't know exactly. We cannot say for the moment. I don't have enough data evidence that to distinguish this reaction and this reaction. The paper, as a published paper or a review paper, indicate that in parallel these two reactions may occur. But yeah, it's possible that. But we cannot exclude one of the reaction. But probably this is actually occurring. Okay, but we cannot distinguish, deny this reaction occur before this reaction. So quite detailed. It's difficult to.
Before slide and this slide, both headings are same. No. This one, no, this one. Next.
Sorry.
Gerani and geranyl both are same or different?
Geranyl reductase, oxidoreductase.
The headings are they both same? Above?
Geranyl reductase. CHLP. That's it. This is the name of the enzyme, and this is name of the enzyme gene. CHL is involved in the gene involved in the chlorophyll synthesis. So usually biochemists assign the enzyme and name it as a granule reductase or granule oxidoreductase, and then later gene or mutant will be isolated, and then they name later they name that the genes involved in chlorophyll synthesis renamed as CHL, ABCDEFGH, or something like that to make things easy to understand. So sometimes naming of enzyme and also naming of the gene or gene product. This is uppercase C is proteins, and the lowercase c is the gene name, and italic is gene name. So this changed. So sometimes molecular biologists dominate biochemists, and then they rename, ignoring the enzyme name. So, but sometimes we have to write both. That's more kind. It doesn't say anything. It may be related to chlorophyll synthesis, but GGR is granule reductase, so it's clearly show the function. So it's better to write both for readers. It's very kind. But it's sometimes people don't do that. So I'm a bit confused. Sorry, CHL, but this is name, and this is more recent, more later, named more later than original. This happens many times, so just don't get confused. Always the name of the enzyme and the name of genes and the gene products are not the same, but it's the same, meaning the same. Okay, so now.
Takahashi, I have some questions.
Okay.
Not question.
Not question.
Very nice talk.
Oh, thank you very much.
I have listened. Yeah. And a lot of detailed things, so whatever you have done, your research about me. Technically, actually, I am looking for the feasibility of these type of experiment in postgraduate level. So, in that context, what amount of your chlorophyll is required for your radioactive experiment?
The pigment composition.
You told it's a small.
Pigment composition by HPLC, you need only small amount, about 50 microgram chlorophylls. It's very volume is smaller better, because it's if you want a separation better, you apply the HPLC a small volume. It depends on the loop. For example, 100 microliter or 50 microliter. Yeah, the lipid determination of the lipid composition is, if you have a good HPLC, it's not difficult. But detection system, if you need diode array, if you can measure the absorption spectra at the same time, it's easy to assign the band, because band is not always the same position. Depend on the sample or depend on the solvent. It's relatively changes. But if you want to separate protein, also you don't need much. For immunoblotting, you need a very small amount, because Western blotting is now very sensitive if you use ECL detection, so very low level, even lower than pigment analysis. But if you want to separate the chlorophyll protein complexes, it's better to see the band, green band. If you don't see, no, if you're working with it, if you don't see band, very discouraging. But of course, for analysis of protein, you don't need much. But in this case, you need a more sample, 100 or 200 microgram chlorophyll at least. But it's still relatively small amount. It depends on the size of the tubes. So, but from the plants, sometimes it's difficult to get enough sample. The Chlamydomonas is a liquid culture. You just need increase the volume of culture. It's easy. But plants, sometimes it depends on the size. For example, Arabidopsis, you have to make many. But Chlamydomonas is easy. That's why the Chlamyd is advantage. You don't need much space. You can save money and save space, but of course, sometimes we need to know what happened in plants, vascular plants, or very useful plants. Chlamydomonas is not useful, so some people criticize. Chlamydomonas is not so. You may eat, but you don't want to eat. So that's. But still, Chlamydomonas is for the basic science, plant physiology, plant biochemistry. This is very rapid.
Yeah,
rapid.
Single cell.
Single cell and rapid, so you can grow the cells in a few days. But in the plant, you have to wait weeks, some months, but it's very fast and quick. So there's a disadvantage and advantage, but it's a basic. If once you analyze the basic phenomena using a Clamminus, and then something interesting things that you can apply for use for the plants. That's good. So both some people works both Chlamydomonas and Arabidopsis. So that is.
Thank you, sir, for the wonderful talk.
Okay.
It will be informative. If you have any questions, you can.
Yeah, welcome to come to me to ask questions.